The overall goal of this experimental setup is to study the fate of trace metals under sulfate and/or iron-reducing conditions induced by microbial activities.
This method can help answer key questions in the biogeochemistry field, such as interactions between key biogeochemical cycles like mercury and arsenic. The main advantage of this technique is that, due to the continuous water flow, the dynamics of the biogeochemical reactions and mobility of trace elements are dissociated and accessible through the sampling ports.
Also, this method can provide insight into the fate of mercury and arsenic, as demonstrated here, in link to iron oxides. It can also be applied to other trace elements or carrier substrates.
Demonstrating the procedure will be Beatrice, a technician from our laboratory.
To begin this procedure, add 500 milliliters of ultra-pure water to a glass reactor equipped with a magnetic stir bar. Add 50 grams of iron 3 chloride hexahydrate while stirring. Manually add approximately 50 milliliters of a 10 molar solution of sodium hydroxide to begin precipitation of ferrohydrate. Next, adjust the pH to 6 and continue stirring for 1 hour to stabilize. For arsenic-spiked hydroxides and oxyhydroxides, prepare 100 milliliters of arsenic trioxide at a concentration of 10 grams per liter. Then, add 70 milliliters of this solution to the iron oxide solution. Continue stirring for 3 hours. After this, centrifuge at 2000 G for 20 minutes. Remove the supernatant and resuspend the pellet with 50 milliliters.